Okay, so welcome back to this next video in which we are discussing cyclic AMP signaling. Okay, so in the last video what we looked at is the structure of the 9 mammalian adenylyl cyclases which are attached to the plasma membrane. Okay, and we saw that they all have these 2 transmembrane domains which are clusters of 6 membrane-spanning alpha helices. Okay, and in between those 2 transmembrane domains you then have the C1 loop, which contains the C1a domain, which forms part of the catalytic domain. Okay, and then after transmembrane domain 2, you have this C2a domain, okay, which is part of this C2 domain, and I should have put that on this picture actually. Okay, so the portion after transmembrane domain 2, this is called the C2 domain. So C2a along with C2b, together they're called the C2 domain. Okay, now C1a and C2a, they dimerize together and make the actual catalytic domain of the adenylyl cyclase. So it's this C1a, C2a dimer which is actually capable of taking in an ATP molecule and converting it into a cyclic AMP molecule and a pyrophosphate molecule. Okay, and as I said in the previous video, there's still a little bit of controversy over whether C1a and C2a are dimerized all the time, even in the inactive adenylyl cyclase. The emerging viewpoints that is growing in its popularity is that they are permanently dimerized together, okay, even when they're inactive, and that to activate the C1a, C2a dimer, all you need to do is cause a little conformational change there, and then it will be put into the active conformation. The alternative view is that the C1a and C2a domains are not pre-dimerized. In fact, in the inactive adenylyl cyclase, they are separate, basically. Okay, and then to activate the adenylyl cyclase, what you have to do is dimerize the C1a and C2a together. But as I say, the pre-dimerized view is gaining popularity. It's the main view now. Okay, right, but it doesn't mean that the other one couldn't turn out to be correct, so be aware that there is that possibility. Okay, right, so what I want to do now is talk about the 4 different families of adenylyl cyclase enzymes. So we've talked about how there are these 9 plasma membrane adenylyl cyclases here. Okay, they are grouped into 4 families, and this grouping is actually helpful because at the moment we've just got a list of 9 adenylyl cyclases, okay, with no real idea what the difference between the 9 is. I've said they all have the same basic structure, okay, but grouping them into these families is actually going to be helpful for understanding the difference between the 9 different adenylyl cyclases. Okay, right, so let's do this now. So I'll do this down here. Okay, so let's start off with group 1 adenylyl cyclases. Okay, now group 1 adenylyl cyclases are also known as the calcium-stimulated adenylyl cyclases. Okay, and this includes 3 members. Okay, specifically adenylyl cyclase 1, adenylyl cyclase 3, and adenylyl cyclase 8. Now we will come back to these right at the end, okay? So when we come on to discuss how calcium can activate adenylyl cyclases, we will discuss adenylyl cyclase 1, adenylyl cyclase 3, and adenylyl cyclase 8 in more detail. Okay, now I should just say before we go on with this, that all 9 adenylyl cyclase enzymes can be activated by G-alpha-S. Okay, and we will come on to that later on. The groupings into these 4 different groups, they're based on other things which can modulate the activity of the adenylyl cyclases. Okay, so these group 1 adenylyl cyclases are the ones which are activated by calciumans. Okay, specifically calciumans bound to calmodulin molecules, but we'll come back to that later on. Okay, right. Now, I'm dropped the Roman numerals now, and I'm just using normal numbers for the adenylyl cyclase numbers. Okay, right. So that's group 1 adenylyl cyclases. Let's now go on to group 2 adenylyl cyclases. So the group 2 adenylyl cyclases are what are known as the G-beta gamma stimulated adenylyl cyclases. Okay, and these are actually going to be stimulated by the beta-gamma complexes of heterotrimeric G proteins. However, it's slightly more subtle than the G-beta-gamma complex just being able to stimulate the adenylyl cyclase on its own. Really, what the G-beta-gamma complex is capable of doing is potentiating the activation that GS gives. Okay, so if the adenylyl cyclase has been activated by G-alpha S, then the G-beta-gamma complex can activate it even more, but alone the G-beta-gamma complex isn't enough to activate these adenylyl cyclases. And again, we'll come back to this right at the end when we come on to discuss alternative ways of modulating the adenylyl cyclases. Okay, so which adenylyl cyclases are in this group 2? Okay, so they are adenylyl cyclase 2, adenylyl cyclase 4, and adenylyl cyclase 7. Okay, so 2, 4, and 7 are in group number 2. Okay, next up is group number 3. Okay, and group number 3 contains 2 adenylyl cyclases. Okay, and the members in group number 3 are the G-alpha I and calcium inhibited adenylyl cyclases. So G-alpha I, and then we'll put / calcium inhibited, and both the adenylyl cyclases in this group are inhibited by both G-alpha I subunits, okay, and also calcium, and we'll discuss both of these things later on. Okay, so we'll see how G-alpha I can inhibit the activation of these adenylyl cyclases by G-alpha S, okay, and also how physiologically relevant concentrations of calcium can inhibit these adenylyl cyclases. Okay, so the adenylyl cyclases in group number 3 are adenylyl cyclase number 5 and adenylyl cyclase number 6. Okay, so that just leaves one left to go in group number 4. So we've had 1, 2, 3, 4, 5, 6, 7, 8. The last one is number 9. Okay, and this goes into group number 4 on its own. Okay, so group number 4 contains the one and only adenylyl cyclase, which is forskolin insensitive. So group number 4 is also known as the forskolin insensitive adenylyl cyclases. Okay, now we'll come on to forskolin, which is often abbreviated as FSK. So this stands for a molecule called forskolin, which is a very, very powerful experimental drug. Okay, it's not used clinically for anything. It would most likely kill you if you took it actually as a medication, but it's an extremely important experimental drug for activating adenylyl cyclase enzymes, and we'll discuss how it activates adenylyl cyclase enzymes later on. Now, it's capable of activating most plasma membrane adenylyl cyclase enzymes, but the one that doesn't respond to forskolin is adenylyl cyclase 9, and that's the characteristic that puts it into this group 4 on its own. Okay, right, so forskolin insensitive adenylyl cyclase is the only one of those is adenylyl cyclase 9. Okay, now I'll stress again, forskolin is an extremely important experimental drug used to activate adenylyl cyclases artificially, basically, okay, without needing to activate G-alpha S. Okay, right. So, there are these 9 mammalian plasma membrane adenylyl cyclases. Most cells have at least one of these expressed. Most cells will have more than one of them expressed. They won't have all 9 expressed, but they'll have 1, 2, maybe 3 of these expressed in their plasma membrane, basically. Okay, and we've seen now how the adenylyl cyclases are regulated by different things and how potentially that will have consequences for what can generate cyclic AMP in those cells. Okay, so for instance, if your cell expresses adenylyl cyclase 5 and adenylyl cyclase 6, for instance, cardiomyocytes express adenylyl cyclase 5 and adenylyl cyclase 6, then these adenylyl cyclases will actually be inhibited by calcium. Okay, whereas if the cell expresses adenylyl cyclase 1, 3, and 8, it will actually have its adenylyl cyclases activated by calcium and therefore will produce cyclic AMP in response to calcium. Okay, so there are important differences between the different isoforms of plasma membrane adenylyl cyclases. They can be regulated in different ways. Okay, and what we're now going to turn our attention to is the mechanism which regulates all of them, okay, the mechanism by which you can stimulate all 9 of these mammalian plasma membrane adenylyl cyclases, which is through the GS cascade. Okay, so we're now going to talk about the GS cascade. So I'm going to start off with a basic discussion of G protein coupled receptors, heterotrimeric G proteins, just to set the scene. Okay, right, so basically, G protein coupled receptors, which are often abbreviated as GPCRs, are an enormous great family of receptors in biology. Okay, in humans alone, we have over 800 G protein coupled receptors, and I suppose I should probably write out its name in full at least once. So G is for G, P is for protein, and then the CR is for coupled receptors. Okay, so G protein coupled receptors. Right, so we have over 800 of these things in humans alone. I think the number that we found overall in all forms of life now stacks up to over 30,000. So there are a lot of G protein coupled receptors. Okay, now what's the characteristic feature that all G protein coupled receptors share? Well, if I draw the plasma membrane here again as these 2 parallel lines, okay, this being the outside of the cell, the extracellular fluid, and this being the cytosol, then G protein coupled receptors all have a characteristic membrane spanning topology where you have 7 membrane spanning alpha helices. So the amino terminus of the peptide is extracellularly and the C terminus is intracellularly. Okay, so like adenylyl cyclases, G protein coupled receptors are a single polypeptide. Okay, and this polypeptide spans the membrane with 7 membrane spanning alpha helices, here in red. Okay, now basically they are a receptor, okay, so ligand molecules bind to the G protein coupled receptor and cause it to change conformation so that it can now cause effects intracellularly. Okay, and I've said that there are 800 different G protein coupled receptors in humans. They will all bind to different ligands, basically. Okay, now, once the ligand is bound to the G protein coupled receptor then, and cause this conformational change, what then inside the cell can the G protein coupled receptor do? Okay, well, basically it can activate things called heterotrimeric G proteins. So let me just extend the membrane up so that I can draw you a heterotrimeric G protein. Okay, so heterotrimeric G proteins. The name tells you a lot about these things. Trimeric tells us that they are 3-membered structures, okay? They're made out of 3 separate subunits. Hetero shows us that the 3 different subunits that make up the heterotrimeric G protein are different. Okay, so heterotrimeric G proteins are constructed out of 3 different subunits. Now these 3 different subunits are called the alpha subunit, the beta subunit, and the gamma subunit, and I'll show these here. Okay? So this one that I'm showing here is the alpha subunit, here this is the beta subunit, and here this is the gamma subunit. Okay, I'll colour them all in different colours. So we'll have the alpha subunit here in red, we'll have the beta subunit here in blue, and then we'll have the gamma subunit here in green. Okay, right. Now, heterotrimeric G proteins then, for short, are often abbreviated to G, alpha, beta, gamma because they have an alpha subunit, a beta subunit, and a gamma subunit. Now, heterotrimeric G proteins can be in 2 states, okay? They can be in an on state, okay, in which the alpha subunit has GTP bound, yes, that is the on state, in which the alpha subunit has GTP bound and it's not bound to the beta-gamma complex. Okay, so in the on state, the alpha subunit has GTP bound, okay, and it will be separate from the beta and the gamma subunit. Okay, however, the state that I've shown you here is the off state. And in the off state, the alpha subunit has GDP bound to it. Okay, so here's the GDP. So alpha has GDP bound. And when it has GDP bound, it can bind to the beta and the gamma subunit. Okay, now the beta and gamma subunit never ever come apart. Okay, so they are often referred to as the beta-gamma complex. Okay, and for short, you can abbreviate the beta-gamma complex down to G beta-gamma like so. Okay, so even when the heterotrimeric G protein is in the on state and the alpha subunit has GTP bound to it and therefore is dissociated from the beta and the gamma subunit, the beta and the gamma subunit remain together, and therefore that's why it's called the beta-gamma complex. Okay, now the heterotrimeric G protein subunits are not integral membrane proteins, so they're not within the membrane. However, they are attached to the underside of the phospholipid bilayer by lipid moieties. Okay, so the alpha subunit has a lipid moiety attached to its which anchors it to the underside of the phospholipid bilayer. Okay, so these structures that I'm drawing here, these are lipid moieties, basically. And the basic idea here is that what you do is you attach onto your protein, whether it's the alpha subunit or the gamma subunit, you attach onto it a lipid molecule, a molecule that is really hydrophobic. Okay, so it's got a really long hydrophobic tail, which isn't going to interact with water at all well. Okay, and the idea then is that this lipid moiety can then implant into the inner leaflet of the phospholipid bilayer, and then it holds the protein, whether it's the alpha subunit or the gamma subunit, attached to the underside of the plasma membrane. So that's the basic concept here, that both the alpha and the gamma subunits of heterotrimeric G proteins have these lipid moieties attached to them, which attach them to the underside of the plasma membrane. Okay, so that means that when this heterotrimeric G protein goes into the on state and the alpha subunit cleaves away from the beta-gamma complex, both the alpha subunit and the beta-gamma complex will still be attached to the underside of the plasma membrane because both of them have separate lipid moieties. Okay, so that's what attaches the heterotrimeric G protein then to the underside of the plasma membrane so that it can now interact with the G protein coupled receptor. Okay, right. Now, there are loads of different types of heterotrimeric G proteins because there is not just one alpha subunit of a heterotrimeric G protein, there is not just one beta subunit of a heterotrimeric G protein, and there is not just one gamma subunit of a heterotrimeric G protein. In actual reality, there are absolutely loads of different alpha subunits of heterotrimeric G proteins, there are loads of different beta subunits, and there are loads of different gamma subunits. Okay, and when a G protein coupled receptor is activated by its ligand binding to it, it will change conformation intracellularly so that it can now bind to heterotrimeric G proteins, and each G protein coupled receptor, each one of those different 800 G protein coupled receptors will have a different preference for heterotrimeric G proteins, i.e. it will want to bind to heterotrimeric G proteins with certain alpha, certain beta, and certain gamma subunits. Okay, now the alpha subunit is the one that it binds to most extensively, so that's the most important one in determining which heterotrimeric G proteins a certain G protein coupled receptor can activate. Okay, now, the different heterotrimeric G proteins have different downstream signaling properties once they're in the on state. Okay, and this is mainly because of the different alpha subunits. Okay, so I've said that there are lots of different alpha, lots of different beta, and lots of different gamma subunits. Okay, the different alpha subunits, when they're in the on state, have very different signaling roles. Okay, the different beta and gamma subunits, it isn't very well understood how they have different signaling roles. At the moment, it seems that it doesn't really matter which beta subunit and which gamma subunit you have in your beta-gamma complex. It seems to do the same thing. Okay, so we're not going to dwell on the different beta and gamma subunits. Okay, it does actually matter slightly for the G protein coupled receptor interacting with the heterotrimeric G protein, because the G protein coupled receptor does bind to the beta and the gamma subunits and does select certain beta gamma subunits over others. Okay, but as I say, the main interaction between the G protein coupled receptor and the heterotrimeric G protein is with the alpha subunit, and therefore the alpha subunit really is the main one which determines whether a certain G protein coupled receptor can activate that heterotrimeric G protein. Okay, right. So, we are going to be interested in two types of heterotrimeric G proteins. We're going to be interested in the GS heterotrimeric G proteins, majorly, and also a very much so related heterotrimeric G protein called G-alpha heterotrimeric G proteins. Okay, now the G here is for heterotrimeric G proteins. The subscript tells you which alpha subunit you have in your heterotrimeric G protein. Okay, so because the alpha subunit is the main one which determines the signaling capabilities of the heterotrimeric G protein when it's in the on state, heterotrimeric G proteins are named after which alpha subunit they have. Okay, so a GS heterotrimeric G protein is a heterotrimeric G protein where the alpha subunit is G alpha S. Okay, and then it doesn't tell us what the beta and the gamma subunit are. They're not important. They're not that important, not compared to what the alpha subunit is. Okay, and a G-alpha heterotrimeric G protein is one where the alpha subunit is G alpha alpha. Okay, now G alpha S is the most famous one, and it's that one that we're going to dwell on most, but I do want to just mention G alpha alpha, because G alpha alpha does basically exactly the same thing as G alpha S. It's capable of activating all 9 adenylyl cyclases. Okay, the reason G alpha S is far more famous than G alpha alpha is that G alpha alpha is much rarer in the body. Okay, most cells in the body have G alpha S expressed and not G alpha alpha. Okay, but certain tissues of the body, particularly the cells of the olfactory epithelium, okay, and that's why it's called olf for olfactory, they have G alpha alpha expressed rather than G alpha S. So it seems as though in certain parts of the body, particularly the olfactory epithelium, G alpha alpha is just expressed rather than G alpha S, and it's just a replacement basically for G alpha S in certain portions of the body. Okay, so I just want to mention that here. We're not going to talk now about G alpha alpha anymore, okay, but be aware that it can do exactly what G alpha S can do, okay, just in certain parts of the body rather than most of the body. So G alpha S is the one that's found in most parts of the body, okay, and it's that one that we're going to dwell on now. Okay, so say goodbye to G alpha alpha now. So basically, a GS heterotrimeric G protein is one where the alpha subunit is alpha S. So I can now fill this in. That's an alpha S subunit. Okay, and we don't know, neither do we really care, what the beta and the gamma subunit are. Okay, so basically what we now need is a G protein coupled receptor which can activate GS heterotrimeric G proteins, i.e. heterotrimeric G proteins where the alpha subunit is G alpha S, okay, to be activated by its ligand. Okay, now let me give you some key examples of GS coupled G protein coupled receptors. So some key examples are the beta adrenoreceptors. Okay, and there are 3 beta adrenoreceptors which all respond to adrenaline and noradrenaline. Okay, and we will be discussing these a lot more later on when we come to discuss some classical GS pathways, okay, and we'll look at how epinephrine or adrenaline can stimulate glycogenolysis in hepatocytes and skeletal muscle cells, and also lipolysis in adipocytes. Okay, so we'll be coming back to these beta adrenoreceptors. But for now, just know that there are 3 of these: beta 1 adrenoreceptor, which is usually just abbreviated to beta 1 AR for adrenoreceptor, and then there's the beta 2 adrenoreceptor and also the beta 3 adrenoreceptor, and they are all examples of G protein coupled receptors which are GS coupled. Okay, so when they bind their ligand, which is adrenaline or noradrenaline, basically that causes a conformational change in those receptors, okay, and that conformational change is transmitted down to their intracellular aspect, particularly these 3 intracellular loops here and the cytoplasmic aspects of the transmembrane helices, okay, and then that will bind onto GS heterotrimeric G proteins. And now what's going to happen is basically when the GS heterotrimer G protein in the off state, i.e. where the alpha subunit has GDP bound and is therefore bound to the beta gamma complex, when this binds to the G protein coupled receptor, what it will do is it will catalyze the release of the guanosine diphosphate from the alpha subunit here. And then what will happen is GTP, guanosine triphosphate, will come in from the cytoplasm and bind to that alpha S subunit. And what that will then cause is a conformational change in the alpha S subunit that then causes the release of the beta gamma complex. Okay, so the alpha S subunit will go into the on state basically, and it will no longer be able to bind to the beta gamma complex. So the beta gamma complex will cleave away from the alpha S subunit. It will also cleave away from the G protein coupled receptor which it had bound to. Okay, so what you'll end up with producing is, if this is the plasma membrane here, you'll get your beta gamma complex now still attached to the underside of the plasma membrane by that lipid moiety. Okay, so here's your beta gamma complex, and I'll just colour these in. So here's the beta subunit in blue, and here's the gamma subunit in green. Okay, and it's still attached to the underside of the plasma membrane by its lipid moiety, and it can go off and have signalling roles, as we'll see later on. It can actually activate the G-beta gamma stimulated adenylyl cyclases, the group 2 adenylyl cyclases, adenylyl cyclase 2, 4, and 7. Okay, it can also have many other signaling roles as well. And then what's also going to happen is the alpha S subunit, once it's bound this GTP, not only does it cleave off from the beta gamma subunit, but it also cleaves off from the G protein coupled receptor which it had bound to in order to get the release of GDP catalyzed by the receptor. Okay, so it will also leave the G protein coupled receptor behind and go off now with GTP bound. And again, it's still attached to the underside of the plasma membrane because it now has that lipid moiety, here, which is, if you care, it's a palmitoyl or myristoyl group usually. Okay, right. So we'll colour in our alpha S subunit here in red. Okay, so basically we now have these two components which can now go off and have signalling functions. The alpha S subunit in the on state with the beta gamma subunit also on its own now. Okay, right, so we're going to chuck the beta gamma subunit aside now, and we're going to look at how the alpha S subunit with GTP bound can activate adenylyl cyclase enzymes. Okay, and I'll stress again, it can activate all 9 adenylyl cyclase enzymes. Okay, so how does the alpha S subunit with GTP bound to it activate adenylyl cyclase enzymes? Okay, well let me draw this. So here is the plasma membrane. Let me now draw an adenylyl cyclase out back again. Okay, so here is that transmembrane domain 1 with the 6 membrane spanning alpha helices. Here is the C1a domain, the C1b domain, the transmembrane domain 2. Okay, then we're assuming that the C1a and the C2a domain are pre-dimerized. And then we've got the C2b domain after the C2a domain here. So this is C2a and this is C1a here. Okay, right, so let me colour the 2 catalytic portions in. Okay, so these are dimerized together, but at the moment they're in a conformation which is not active. Okay, so they're not actually catalyzing the conversion of ATP into cyclic AMP and pyrophosphate at the moment. Okay, now, what's basically going to happen is our alpha S GTP subunit can come and bind to this dimer here, the C1a, C2a dimer, okay, and can promote a conformational change in that C1a, C2a dimer that can then cause it to become an active conformation basically, and then it will be turned on and it can now start converting ATP to cyclic AMP and pyrophosphate. Now, as I said previously, this is still controversial, okay? The alpha S subunit with GTP bound to it is actually capable of binding to both the C1a domain and the C2a domain. So it binds to a little portion of the C1a and the C2a domain. And the people who are in favour of the theory where the adenylyl cyclase C1a and C2a domains are not pre-dimerized, say that the alpha S subunit works by promoting the dimerization of C1a and C2a. Okay, so in their theory, what happens is C1a and C2a aren't dimerized together in the inactive adenylyl cyclase, and then if you want to activate the adenylyl cyclase, what you have to do is promote the dimerization of the C1a and the C2a domain, and alpha S GTP does that by binding to the C1a domain, binding also to the C2a domain, and forcing the two together basically, and therefore assembling you an active adenylyl cyclase. Okay, but as I say, the leading theory now is that C1a and C2a are already dimerized together, and the alpha S GTP just binds to that pre-dimerized C1a and C2a domain, and then causes a conformational change that then promotes it taking on a conformation that is actually capable of catalyzing the conversion of ATP into cyclic AMP and pyrophosphate. Okay, now, before we then go on to have a look at what the cyclic AMP is actually going to do, what I just want to mention is how this drug forskolin can activate adenylyl cyclase 1 to adenylyl cyclase 8, but remember, not adenylyl cyclase 9. Okay, so forskolin, often abbreviated as FSK. Okay, and it comes from plants, this drug, and basically what it does is it manages to bind right at the interface of C1a and C2a, and again seems to cause this conformational change that activates the C1a C2a dimer, causes it to assemble into a conformation where it's now capable of actually catalyzing the conversion of ATP into cyclic AMP and pyrophosphate. Okay, in the other view, where the C1a and C2a domains are not pre-dimerized, then the forskolin is believed to promote again the dimerization of C1a and C2a. Okay, so remember that forskolin is also capable of activating most adenylyl cyclases except adenylyl cyclase 9, and therefore also is capable of beginning the cyclic AMP signaling pathway. Okay, right, so I think we'll call it there for this video, and in the next video what we'll then look at is what the cyclic AMP, once it's generated, is actually going to do, and we'll start by looking at the principal target of cyclic AMP, which is protein kinase A.